Welcome back, everybody, to Bio 2022 here in San Diego. I'm Greg Slobodkin, Editor-in-Chief of ScienceBoard.net, and I have the privilege of being here with Ashley Zehnder, who is CEO and co-founder of Fauna Bio. Welcome.
Correct. Thank you. Yeah, thanks for having me. I appreciate it.
I have to say, I read your bio, and it is the most interesting science background I think I've ever seen.
I am somewhat unique that way, yes.
Definitely. So I don't even know where to start, but I want to throw this out there.
Okay.
You are a veterinarian.
That's correct.
And you're also a
Cancer biologist.
cancer biologist.
Yep.
And obviously you're the co-founder of a company, a startup. So talk to me a little bit about Fauna Bio, but within the context of your background.
Yeah, it was interesting, Greg. So I started out as a clinical veterinarian doing an exotics residency at UC Davis. And what I noticed during my residency is that there were different ways that animals would present with different diseases. Particularly, there were species that got a lot of cancer and some that almost never we saw with cancer. The problem is that not a lot of veterinarians do like molecular biology training during their veterinary career. A lot of medical students will do rotations through labs. Very rare for vet students to do, more and more now. And so I was really frustrated by the lack of understanding of the molecular mechanisms of the diseases that we were seeing in my patients, right? So I ended up having a chance to do a cancer biology PhD at Stanford. There was a program specifically for vets who wanted to do PhDs. I was the third vet at Stanford to do that, and really worked on human cancer throughout my PhD. But during the process of getting into that program, I was interviewed by really preeminent cancer researchers, and they looked at me in my CV, which, as you notice, is a little bit unique, and said, "Why are you here?" essentially. And so I told them about my patients, right, that I was treating with radiation and chemo for their different kinds of cancers. And I want to understand what was shared between humans and animals for these diseases. And the man looked at me and said, "Birds get cancer?" And I was like, "Well, they have cells." Like, I mean, why? It was a logical question for me. But this was a man who studied cancer and only thought of it in the context of humans and mice that he gave cancer to, not any other context, right? So I realized that there was actually this much bigger gap in biomedical knowledge, which is the fact that this is shared physiology across species, but biomedical research largely only considers the human condition and not what we can learn from how these diseases present across species. So while I did a cancer biology PhD, I was fortunate enough during my postdoc to meet my two co-founders. So Linda Goodman, who has a human genetics PhD, got really frustrated trying to figure out risk variants for human cardiovascular disease, looking at just human data. We're just honestly too similar to separate signal from noise. So she went to comparative genomics and said, well, what if I look across 200 or 300 genomes? Can I tease out those genes that are really highly conserved and thus more likely to be functional in disease? And our CSO is Katie Grayback. She has a human genetics PhD. She rotated through a lab of Dr. Sandy Martin, who studied 13-line ground squirrels, saw the temperature trace that I just showed you of hibernating 13-line ground squirrels. Their body temperature literally does this during the hibernation cycle. And she's like, this is way more extreme than anything that humans do. I want to study that. And so she built a biobank of samples during her time as a PhD student of really precisely timed tissue samples from every tissue in the 13-line ground squirrel. And we in-licensed that as founding IP at Fauna. So that's a physical biobank that resides now in Emeryville, California, that we use for a lot of RNA-seq studies. And we use that alongside this very large collection of 452 now whole genomes from different species, including humans, that we use to find novel targets for human diseases. So it's really just about using all this animal data to really highlight where the important nodes are for disease biology. And we made this analogy earlier, if target discovery is trying to find a needle in a haystack, which people use that a lot, AI and ML people are using to really filter the hay faster. And we say, no, just use a bigger magnet, right? Just use the right data to find the impactful targets.
So you mentioned AI and machine learning, and I want to get to that.
Sure.
But you mentioned hibernation.
Yeah.
And so talk to me about the applicability of hibernation biology to humans and human diseases.
Yeah, it's really interesting. And I didn't appreciate this because even as an exotic animal veterinarian, not a lot of people keep hibernating animals as pets, so I never really treated them as a veterinarian. They're highly adapted to protect their tissues from damage. You've got a 100-gram animal that goes down to nearly freezing and stays there for most of the winter, but then warms up through these really acute periods of rewarming called interbout arousals. They get this rush of oxygenated blood into every tissue in their body, brain, heart, kidney, liver, that actually can be really damaging. And so they've developed ways to not only resist damage during the whole hibernation cycle, but if they are damaged, they can repair this more easily. So what we look at are really markers of disease resistance. So where they've started to turn on collagen and fibrosis, where do they turn it off? Where they've started to have tauopathy or a tau phosphorylation in the brain, where do they turn it off? They have changes in their neural connectivity when they're in the depths of interbout arousal. They're actually brain dead. Their neurons are tracked. They are disconnected. They reconnect every interbout arousal, so every 1 or 2 weeks. It's just crazy physiology. It's done using the same genes that you and I have, but in just more extreme ways. And so we can use that extremity to find solutions for things like Alzheimer's disease and heart disease. We're working with Novo Nordisk on a program for obesity. These animals double their body weight in the spring and then slowly use it over that time course. They increase their metabolism 235-fold in an hour, right, through these interbout arousals. There's literally nothing in the human condition that compares to that, but we can learn from that to say, how do we increase human metabolism for patients that have lost a significant amount of weight, have reset their metabolism to a low point, and then regain it because they can't reset their metabolism. Well, ground squirrels do it every 2 weeks, right? So it's a lot of these kind of physiological observations that allow us to link to human disease. We used to do that by picking Katie's brain. We do it now because we built a knowledge graph. We just kind of released a press release yesterday about this, named Centaur. Centaur takes expression data from these animal models that we work with. It links it directly to human disease data, DisGeneNet, UK Biobank, dbSNP, all these human databases that exist, and says, oh, look, there are these genes going down in the ground squirrel. This links directly to a human disease. We should look at that network more closely. Oh, and by the way, there's a drug that targets this gene that's been repurposed for something else. Wow. So it links all of that together automatically, and it removes us having to ask Katie all the time what we should be doing with some of this animal.
That's incredible. So where do you go in terms of clinical trials, and where do you see the technology going in its evolution? Yeah,
so like I said, I mentioned we have a partnership with Novo Nordisk. We also have our own internal drug development pipeline. So we have a compound that we developed by looking at expression signatures and hibernators. We can then match that to compounds. We do also have some novel target programs ourselves. So the idea is to really get these programs far enough into a late preclinical or early clinical stage to be able to partner them for tractable indications while we broaden into other disease areas. So aside from those kind of top lead programs, we have emerging discovery programs now in GI disease, so IBD. We're starting to look at renal complications. We have several programs in fibrosis around the heart and other organs. So it's really just a matter of starting these programs, getting tractable targets, validating them, and then taking them far enough along that we can then partner them strategically where it makes sense.
So you talked about when animals hibernate, it's such extreme conditions where the tissue actually gets damaged.
Mm-hmm. Yeah.
But it sounds like there is a regeneration mechanism as well.
Yeah.
Can you talk a little bit more specifically on that?
Yeah, there's definitely evidence in the tissue histology and also in the transcriptomics of looking at healing and repair mechanisms. How animals are able to reverse, like I said, things like collagen, that if you and I had a heart attack and laid down collagen in your heart, it doesn't usually go away, right? So these animals are able to reverse those types of pathologies. We're starting to look now into other species, not just hibernators, but there's other models. One is the spiny mouse that can literally regenerate things like a severed spinal cord, right? They're even beyond what we can do with the hibernating mammals. So I think it's really about understanding the physiology and then understanding the human disease question and saying, what is the right animal to answer this question? Mm-hmm. It's probably not a mouse. It's probably something else. And asking the right questions of the right models, right? And that's really where we focus. Yeah.
I mean, people point to this all the time. I mean, mice models are the popular platform.
Sure.
And yet, I mean, to be able to make that leap from what happens in a mouse, will it actually behave the same way in a human?
Yeah.
There's a disconnect there.
Of course.
I mean, so as a veterinarian, as someone who understands both species, I mean, what do you think that the answer is there?
So I think there's never one model to answer the question. I think you have to use a lot of different data types. What we do is we first validate everything that we do in human cells. So human primary iPSC-derived cells, primary human cell lines. If it doesn't work there, then we sort of move on, right? I don't really care if I can cure squirrels of heart attacks, right? So it really has to pass that filter of being functional, a human cell assay that mimics a human disease. We also bring in several different types of human, either genetic data. We curate a lot of functional studies. So RNA-seq studies of humans in heart failure, RNA-seq studies of humans in obesity or NASH. And we bring that data in, we clean it because it's really messy, and put it on the platform and use that to validate the signatures we get from the ground squirrel. So there's this huge human data filter that everything has to go through before we move into any more traditional preclinical models. And so you have to kind of use a balance of those things, right. And nothing is perfect, but you try to use multiple evidence points to prove that what you're going to do is hopefully going to work. So.
Yeah, I mean, and I think about your analogy about needle in a haystack and your AI platform and trying to really get more precision.
Yeah.
And I think earlier you were also talking about, it doesn't take as many samples.
It really doesn't. Yeah, that's the interesting thing. We went back and kind of did this post-hoc analysis of like, well, okay, how much data did it take for us to start these discovery programs that we've now validated in vivo animal models of disease? Mostly it took about 60 samples, right? And so you think about starting a target discovery program with humans, you could never do that with 60 samples. And that was like a super ultra rare disease in like one family. And these are more complex. This is heart attack models, right? You could never do that. And so I think it's about the extreme nature of the data, using the transcriptomics, and using that in a very precise way. You don't actually need petabytes of data, as some drug discovery companies like to talk about. In some cases you do, because you don't have the right models to look at that. But you can actually do it with a very small sample set. And actually we're hiring our first platform developer, bioinformatician. He was used to looking at gene expression data for human prostate cancer. He was used to working with hundreds and hundreds of patients, sometimes thousands of patient samples. We said, we have like 60. He was like, it's not gonna work. We're like, just try it. And we gave him the data and he played with it and he was like, wow, this is really clean. I'm like, we know. And so he leads our platform development. We converted him to Fauna Bio. But he didn't believe it was possible. It's like, oh my God, you can do it. I'm like, yeah.
Well, it's been fascinating talking with you.
Thanks for inviting me. I really appreciate it.
So, we'll be watching and seeing what develops with Fauna Bio, everybody. So thanks again, and stay with us for continuing coverage of Bio 2022 here in San Diego.
Thanks so much.